Let's get oral. Let's get oral.
Hello. It's Jonathan and Camille's show at Let's Get Oral today. And I'm so excited to sit down with Dr. Laura Weyrich because she's another microbiologist. And boy, do we love having her on this show. She has been doing some really incredible work, both historically and also globally. She runs 2 labs, one here at home in Pittsburgh and one in Australia. And she's been at it for many, many years, has a deep repertoire to pull from. And currently she's running a microarchaeology lab where she's been digging into Neanderthal plaque and evaluating kind of what a good microbiome looks like from a historical perspective, and then moving that forward to practical interventions. So looking at oral microbiome transfer, very exciting work. And she completed some caries work, cavity, by using that method in animals. And now she's moving to periodontal work, also in animals, but with the same principles. So we're going to dig into that. But she's basically bringing us this historical analysis on dental calculus to reconstruct what we think a current oral microbiome could look like in a healthy setting. So we're going to dig deep with her on this subject. And I'm going to turn to you, Laura, and ask you, how did you get involved in the oral cavity in the first place? It's an unusual niche that we're sitting in here.
Yeah, it is, but it's one that needs more study. So
Oh,
that
my
was
goodness.
one of the things that attracted me to it. I did my PhD in infectious disease, and I was studying a pathogen which causes whooping cough in the lungs. And so I was already kind of looking at the respiratory tract and doing a lot of work in the microbes that live in that part of the body. Well, everyone else, I think, was focused on the gut. And,
Yeah.
I'm so proud of it and just amazed of all the work that's happening in the gut. But what I saw at the same time was a real lack of microbiome research in the mouth. And, we treat it so differently in the medical sphere. The medical and dental feel so different, and it's no different in the microbiome sphere. It's like
Yeah.
the mouth is separate from the gut. That same ideology was being kind of transplanted into my field. And I thought, why are we doing this, right? Like, we need people who work on the mouth. And so,
Yes.
yeah, no dental background, but working on these microbes that I think are super critical for our health and have been really critical over evolutionary time.
Yeah, absolutely. And bringing up the gut, if you go into those studies, how are they inducing changes to that gut microbiome? Through the mouth, right?
Yes.
Which is just the most ironic part of this discussion, right?
Yes.
A frustrating, we're just going to ignore how it got there and just study the gut alone as
Exactly.
all of medicine.
Exactly.
Okay, so with your historical perspective, studying Neanderthals and also, we'll say, communities that haven't been westernized, what are some of the learnings that you could bring forward, some illuminating facts that you've seen in these populations?
Yeah. There's 2 that come to mind straight away. The first one is that Neanderthals had amazing oral health. They had like perfect teeth, right? I always tell people, if I could have teeth as nice as Neanderthal, I absolutely would.
Yes.
There's a few populations where they had, incidents of cavities or caries or decay, whatever you want to call it. But for the most part, they had excellent oral health.
Could you pause there and say, maybe were there some environmental factors that were at play for those cavity-ridden samples?
Yes, probably some dietary factors. And so, in the Paleolithic into the Mesolithic, the places so far where we've seen populations that had, bad cavities, they were places where they were eating lots of carbohydrates. So one site in particular, they ate a lot of acorns. And so, we don't eat acorns today, but that if you boil those down and whatever, you can get a really nice flour with lots of, carbohydrate in it. And so that was a place where, archaeologists found lots of cavities in people. And when I say Neanderthals, I also refer to them as people. I think they're kind of part of our ancestral network. And so, or our fiber, I guess I could put it that way. So and the same is true for anatomically modern humans. Some of those early sites where we see them, we assume that they're eating lots of carbohydrates, that it's that diet that probably is different. But I will say there's just such a limited amount of information on people who lived, more than 10,000 years ago. There's
Yeah.
just so few individuals that have been found to study, and we're limited to sort of bioarchaeological study. And, my field's putting DNA on top of that and putting proteomics on top of that, but it's just so, so limited. There's handfuls of Neanderthals that have calculus, for example, that we can study. So, maybe with more study and as we go throughout the years, we'll have a lot more to say about that and understand, what bacteria were involved in some of those processes. But right now, at least, we think that it's this environmental dietary shift that probably causes that.
Interesting. In those plaque samples that are so old, I took a bacterial paleontology class when I was in my grad school days, and I absolutely loved it. But I know contamination is like a huge argument in the field. It's really hard to kind of control. And so how do you kind of parse that out?
Yeah, that's an awesome question. And that's a problem, I would say, even in studies of people living today. And we can talk about that if you want. But in ancient individuals, when you get to sort of perform ancient dentistry on them, right? Like you take your tool of choice. So that can be a dental pick or a small screwdriver if that's easier, or whatever you're going to use, right? And you pry off the calculus. And for us, it needs to come off in a large chunk. Like it can't just crumble into sand or to dust, right? That means it's probably not well enough preserved to get signal out of. And so if it comes off in a chunk, we then can take it back to a lab environment. And we work in these spaces that are, for those of you that are microbiologists, BSL-3 labs, but they're made in reverse. So here we're trying to protect the samples from us rather than us from the samples. But it's the exact same sort of environment, right? You've got, again, it's in reverse. So you've got positive air pressure instead of negative air pressure, but you've got HEPA filtered air. You're in a complete bodysuit where the only thing sticking out is this. And even that you have a plastic face mask over. And so, we really are doing everything we can to keep from introducing any sort of modern oral bacteria into that sample.
Right.
We then bleach that sample. So we take this, super valuable itty bitty tiny piece of calculus and we put it in bleach. And it, the first time you do it, your stomach drops because you're like, oh my God. But that helps to oxidize and destroy the DNA or, microbes or anything that's on the outside of that. We also deal a lot with soil microbes, any archaeologist that sneezed, on that individual over the last 80 years or something, we're also dealing with that, right? Some archaeologists also lick bone from rock to tell the difference. And so,
no, stop it.
Yeah, the texture is very different.
Fascinating.
Yeah, it's weird, super weird. So we have to deal with all of that, right? So the bleach helps get rid of
No, I mean the mouth.
Yeah. Yeah. The things people do that you don't know they do.
That is absolutely wild. I love this.
Yeah.
Adding that to my trivia collection.
Yeah, exactly. How do you tell the difference between bone and rock? You lick it.
Lick it.
Yeah, you lick it. So anyway, we soak it in bleach and that helps destroy all of that. We also then UV treat it. So it's put under a UV linker and that, cross-links any of the DNA so it can't ever be opened or utilized after that. And it's only after that do we then try to dissolve the calculus and break it open and try to see what bacteria are inside. So, and even when we do that, on top of that, we process samples right next to it. We call those control samples where we're looking for any DNA that's just present in our reagents, any DNA that's present in the lab. Sometimes we'll also do swabs of the museum and wherever we're coming from, and we'll sequence the DNA in those swabs to see what bacteria were in that museum environment. So we compare then our calculus, sample and the bacteria in it to all of these environments that the calculus might have been exposed to and make sure that it's completely different. So I've ended up in my career doing a lot on contamination control and contamination avoidance. And, you're never going to get rid of it. You need to monitor it, not put your head in the sand and pretend it doesn't exist. But we've done a lot of that to make sure that these ancient signals we get are truly, truly someone's ancient oral microbiome and not, spit from an archaeologist from 80 years ago.
Oh my goodness, that's amazing. Okay, so now that we have this purified calculus, or clean as it's going to get, as far as kind of our favorite pathogens du jour to blame on oral pathology, do you see any of them in these old historical samples?
Oh my gosh, I love that. Pathogens du jour. Blame things on. Oh, you're preaching to the choir. That's amazing. So the quick answer is yes, we absolutely do. So we see a lot of the red complex, right? So things like Porphyromonas gingivalis, it's
Yeah.
absolutely present. It is there in Neanderthals. They did not
And what, they're healthy? Oh my.
they did not have gum disease, right?
Yeah.
Which
Mhm.
is something else we can talk more about too. It's just the
Yes.
presence of these supposed pathogens in people who are orally healthy, right?
Yeah. Mhm.
Especially from different non-industrialized cultures and ways of life. So we can also see some Tannerella occasionally. And, it's a good question about what egg-like species it is, I would say. We don't necessarily see as much treponema. Usually that is something that we kind of do see more associated with gum disease, but it's there at least in other ancient people. So, ancient farmers and things like that that are generally orally healthy, it's still there. Streptococcus is, and mutants, Streptococcus mutans specifically, has been a really interesting project. We're getting ready to publish a paper on that. That is not found widely until much more recent in time. So that is something that
Interesting.
we think maybe actually is one of these adaptations to agriculture and adaptation
Yeah.
to more carbohydrate in our diets. But we do find other Streptococcus species that are there and potentially, associated with cavities in the mouth, although they're there at much lower proportions. So they're
Sure.
not, highly abundant. So yeah, I think that's one of the things that my research is really kind of grappling with right now is that the presence of these supposed pathogens in ancient people who are otherwise very orally healthy. And so what does that mean, right? Does that mean that their genomes and their functions and their abilities have evolved over time and now they're pathogenic today, right? Or does it mean that, actually it's the environment that might control whether or not these microbes contribute to disease? Actually, it's probably a little bit of both.
Yeah,
A lot of both,
absolutely.
actually.
Mhm. Yeah.
But I think, yeah, we have to be looking at pathogens very differently than how we think about them in terms of like Cox postulates and things like that in the mouth anyway.
Agreed. I think the move to stop calling them pathogens is kind of an initial nod in that direction. So
Yeah.
I appreciate efforts to do that. But yeah, I do think host factors and environmental factors are all at play. And then if we just appreciate the difference from species to species and know that, what is that, like a 87% gene homology is required for a species to species, that's quite huge. And then just subspecies is 99.9, which is us and chimps, right? So there's still quite a big difference between
Yeah.
these subspecies that kind of, I think, goes underappreciated when we're generalizing functions.
Yeah, 1,000%. And then on top of that, these microbes act so differently in the context of other microbes. And,
Right.
everybody has a sort of distinct microbiome, and we're not thinking about how those other microbes may control those pathogens in some ways too. So, I mean, the list is long, right, about these kind of, exogenous factors, these additional factors that might be influencing people's oral health.
So, speaking of exogenous factors, in those calculus samples, can you get an inference of saliva quality from those?
Ooh, that is a great question. We have not tried yet. So we're working on it right now in the
Yeah.
lab, trying to do some metabolomics and metaproteomics. So looking at,
Yeah.
the chemicals and the proteins that are present in that calculus to say, like, can we detect non-bacterial sort of proteins, maybe human proteins, or the like that might be able to help us infer some of that. Yeah, that's really, I would say, like one of the standing questions, and would be great to pair that information with the microbes and really
Right.
have a more holistic sort of view of that. But yeah, I literally have a postdoc in the lab right now today working on that. So we're headed in
Amazing.
the direction. It's not there
I
yet.
love, I mean, all the work, the detail that goes into your work is astonishing, and I'm just so appreciative. Um, yeah, I could be here all day possibly, so apologies in advance.
No, no, I'm not.
Let's see. Okay, so as far as moving forward and kind of taking these pieces into a more therapeutic approach, you've been doing a lot of oral microbial transfer. And some of our audience may already be familiar with fecal transfer, which is everybody wants to cringe, but people who suffer from Clostridium difficile really do well with that supplantation. So, well, just give us a kind of synopsis of where you're at today and some of the key understandings that you're utilizing to move forward.
Yeah, no, that's great. As you mentioned before, we work a lot with indigenous communities, and it's not just here in the United States, it's all over the world. And in doing a lot of that work, we were starting to understand that they carry some of these pathobionts or pseudopathogens, whatever that you want to call them. And they still don't have disease in all cases, right? Whereas in other cases, people didn't have some of those pathobionts and had very severe disease. And so, this ideology that we truly understand everything that happens that underpins the microbial part of these diseases, I think was really frustrating to me. And, at this stage in my career, this was about 6 or 7 years ago, I'd been working with communities, studying all sorts of ancient individuals, working with living communities today. And we were getting this really, I would say, much more detailed picture of these evolutionary signatures that contribute to disease in the mouth. I was talking with my husband I'm so excited about this. And I was talking about the next steps and where I was going to go. And he is somebody who has kind of some of the early stages of gum disease. And, he looked at me, he's like, this is great. This is really awesome, all this work that you're doing. But when are you going to do something that actually cures the disease for people or solves it, right? You've got all this foundational knowledge behind how it works. What are you doing with it? So what, right?
Right.
And, it was one of those kind of light bulb moments, yeah, what are we going to do with this, right? And you always write in grant applications that somebody's going to use this someday to improve health, but it's no, who's going to use it?
Yeah, who's that? Yeah.
Who's that person, right? And, had been very involved in the microbiome research in other areas of the body, helping other researchers in the gut and things like that, very tangentially, even though my own research has been focused on the mouth. And knew about the fecal microbiome. I always say transplant, but transfer works fine too. So the FMT, for lack of a better, an easier way to say it, I guess.
Yeah. Yes.
So I was very aware of the FMT literature. And I thought, why are we not doing that in the mouth?
Yeah.
And at that stage, there was one study published in dogs by a group that was an international group. And they had taken one healthy dog donor and they had taken saliva from that donor and put it into dogs who had periodontitis. And the short of it is they didn't see anything. There was maybe a quick switch of the microbiome for maybe a couple of days. And then by 2 weeks, it was gone. There was no change in disease status, kind of oh, okay, this isn't going to work was the end result.
Did they deplete first? Sorry, just have to ask.
If I remember correctly, they did do some cleaning, but probably not as intense as the cleaning I would say that we end up doing.
Sure. And that would be chlorhexidine, for example.
Yes. Or debridement first. You know what I mean?
Okay.
And that kind of combination. Yeah, I do believe that they were dogs that they'd brought in for cleanings of some way, but I'd have to go back and read the paper. Forgive me.
Sure. I get it. Yes.
We did that. Anyway. And so, I was talking to other people about it, and the general sentiment in the oral microbiome field, was that oral microbiomes don't change.
Right.
You're given those microbes from your mother. Once you've got established, when your permanent dentition comes in, those microbes are static and they're not ever going to change in your whole life. So of course, an oral microbiome transplant wouldn't work. Of course, it's not going to work. And I thought, well, yeah, I know biofilms are very robust to change, right? It doesn't matter if they're in your plumbing or in your mouth or wherever they are, right? Those microbes have an incredibly tight-knit network. They're going to be very difficult to change. But I also know that if you can completely remove them and replace them, you might actually be able to change them. And so it was really kind of thinking about all that and wrestling that. And so I also talked a lot with an oral microbiologist at the University of Adelaide named Peter Zilm, oral epidemiologist dentist at the University of Adelaide, Lisa Jameson. And, we had a lot of coffees and we talked about that. Is this possible to actually change someone's oral microbiome if we think about it differently beyond saliva, right? Sure, saliva is like your partner, right? You're going to kiss them. You might pick up some microbes. There's evidence of that. But your whole microbiome doesn't change when you kiss your partner, right? So saliva is probably not the ticket here. And so what we probably need to focus in on is the biofilm. And so Peter Zilm had been working for a couple of years to develop a 3D printed flow cell that could effectively grow dental plaque in the lab. And so he had some early prototypes, which is wild to me that you can basically 3D print a mouth and, use an artificial saliva to grow dental plaque in the lab. I was like, that's phenomenal. That's amazing. I think we can use that. And so we got this idea to kind of combine the idea of a fecal microbiome transplant and this growth of plaque and turn that into an oral microbiome transplant. So, our idea was to harvest dental plaque from people who are very orally healthy. And we kind of have two populations, right? That can be people who are in dentistry school or in, going to school to be a hygienist who great care of their teeth, right? These are the people brushing their teeth 3-4 times a day. They floss twice a day, doing all of the things to probably have great oral health. And then there's these other people who are phenomenal, who never brush their teeth, never go to the dentist, never had a dental problem in their whole life, right? They eat lots of sugar. Some of them smoke, right? But yet they have perfect oral health. And so we wanted to sort of collect all of their bacteria and be able to study them and understand, what's different across these different healthy populations and what drives that health signature. Is it their saliva? Is that it? And it's completely independent of the microbes, or is it the microbes themselves? What is this? So we got to study them, and we put out a couple papers on that. There's a lot more coming behind it because there's just a wealth of information. But it was 100 of these people who were, very orally healthy. We then worked a lot more on this flow cell. I think we're on version 3 or 4 now, where we can have this artificially designed mouth and grow these dental plaque communities in the lab. We can do microscopy on them. We can do metabolomics. We can do metaproteomics. We can do metagenomics, whatever it is we want to do to characterize them. And so we've done a lot of work to characterize these really healthy microbiomes, figure out how to grow them, and figure out how to reproducibly grow them in ways to then put into somebody's mouth. And then we've been working on doing preclinical trials in animals, and we're gearing up to do one in humans here very shortly to figure out how we then take these microbes from this flow cell, mixing them in some sort of material, right, and then putting them into the donor. And at least in a mouse model, this is highly effective at reducing cavities. So this is kind of where we're sitting.
That's all very exciting. I have so many questions. Okay, on the flow cell, both Jonathan and I have worked on similar in vitro models. We had an artificial mouth lab at Colgate, back in the day.
Yeah.
And both of us kind of evolved some of those models as well. And anyway, so I know host factors play a huge role in how that community of microbes develops. So have you played with that aspect of the show?
Yeah, we've played with a couple. We have stuck with our artificial saliva so that it's always reproducible and always the same. And we know exactly what the components are. Although there's been a lot of talk about whether or not we should be using harvested human saliva to do that.
Yeah.
But we've stuck with that. And then what we've done is play with some of the nutrient content. So we've, for example, added more sugar to mimic like a more carbohydrate rich diet. We have shifted the pH, for example, to see what happens. So in some of those instances, we have very much played with it. There are other ones, though, that I would love to explore more, mostly in like the composition of the saliva and sort of what that looks like. I would love to know like the concentration of amylase and how much that influences, so there's all sorts of questions that remain in that space. And I have a grant right now through the NSF to help look at a lot of those factors and how they shape that flow cell growth and how they might shift our donor microbiota, how they shift their bacteria to a state that is even more healthy,
right?
Or a way that's also more protective. But that's one of the beauties of the flow cell, right? Is you can play with all of that. You can also add in other microbes. You can add in probiotics. You can add in, putative pathogens or pathobionts and see what happens, right? Mm-hmm. And so there's a lot of work that will come down in that space that is actively ongoing right now in the lab.
So one of the factors I was utilizing to increase the inflammatory profile was simply adding a slightly larger percent of serum.
Yeah.
So helping to shift the profile to see how that impacted. I was doing cellular-based assays, but
Yes.
Wonder if those, because that would be a factor for some of those pathobionts, right?
Yes. Yeah. So we actually have done that experiment where we've added serum. And it's in a paper that is about ready to be submitted. I will tell you though, it's a hot mess to try to figure out in the way that we did it. And that is because serum contains so much DNA in the areas that it's processed. So it's sterilized, right? Like it's been sterilized, it's been gamma radiated, whatever it is, however you get it from your developer. But one of the problems is that bacterial DNA is still there and it's present in those. And so trying to get serum without bacterial DNA is like, I would say like almost impossible. Like if you want the components of the serum, intact, like getting rid of all of that, it's very difficult. So we've done that experiment. We did see shifts. The contamination in it is an absolute nightmare, but the serum absolutely does select for sort of growth of pathobionts, like exactly like you would kind of ensure that it would. And so it does raise a lot of questions just about what type of individuals we want to put our technology in and thinking about who might be good candidates for that technology versus who might be, no, we need to get you to, a better place, like in the context of your gum health, and then we can try a transplant.
Right. So what you're implying there is systemic health might be informing the show too much to have an oral microbiome transfer be effective potentially. Yeah.
Or, we have to be also aware of potential risks, right? Like we are transplanting live microbes, into people. Yeah. And so somebody who, has very severe periodontal disease, that might not be a good candidate for that yet, right? right? Like,
Yeah,
whereas if we
right.
can work through classic channels to maybe reduce, some of the symptomology of that disease, then maybe we could think about that, right? Versus preventative care, right? For somebody who might be at risk or is maybe in the early stages of that disease, our technology might be much more well suited for those folks. Does that make sense?
Right. Yeah, absolutely. Okay, so follow up on I know you do a lot of work to prevent virus transfer, for example. So when you're taking plaque and calculus from a healthy donor, you're making sure that you're minimizing all the risk. You're using these flow cells to amplify the right thing, so minimizing that potential viral transfer. But what about phages?
Yeah, it's a great question. So we can still see some phages, right, that come
Okay.
along with the bacteria. We don't in our
Do we
flow cell.
like them? Are they friends?
Yeah, that's actually a really interesting question, right? And yeah, I mean, I think that they probably are, right? I think that
Yeah.
there's a balance that's established, obviously, between those phages and those microbes that
Yes.
keeps them in check. But being able to harness that relationship, I would say, is still a long ways away. And a
Yes.
lot of the other people have tried, with various different levels of success. But in the plaque sort of environment, I think it's very difficult for phage delivery and things like that. But that's a whole nother conversation for another day.
Okay. Okay.
But yeah, so you're completely right. And that we don't have any human cells, for example, in our flow cell. And so we don't see carriage of those human associated viruses. We're going through the process right now to have that third party verified, just to be 1,000% sure that that's all very safe to transplant. But phages are a different story, right? And those phages, in those microbes, as long as those microbes are around, I do anticipate that those phages will continue to stay around. But
Yeah.
understanding their role and sort of maintaining these healthy oral environments, yeah, is definitely something that's very understudied and needs more more people on it for sure.
Oh, absolutely. Could not agree more. Okay, so when we're talking about host factors, I think about, of course, the famous Rob Knight study of transferring mice gut biota into germ-free fish and then vice versa. So you take mice to fish and fish to mice. That's basically what we're talking about. Okay, so then when we do that, that fish microbiome ends up populating like a mouse microbiome and it's put in mice, suggesting that the host factors are really driving the bus, which I don't think is 100% true, but I do think it plays such a big role. I do wonder about, the longevity of these treatments and durability. We saw that in the dog study, maybe 2 weeks is best we get. But I mean, I think that there's room to play here. So what host factors do you think are important? And then how do we support these good changes that we're trying to implement?
Yeah. Yeah, it's a really great question because host factors are important, right? I think the way I think about it though is what it probably means effectively is that you just need to find the right donor for you, right? Like we probably need to be pairing sort of donor and recipient together to make sure that we've got the right set of microbes that might be most successful in that particular recipient. And so we can do things like match people's ethnicity or match people's environment or match people's smoking and drinking habits, right? It doesn't mean that there's no one, healthy out there, right? Like to sort of
Interesting.
find that donor.
Yeah.
So
Wow.
absolutely, I completely agree. I don't think that it's all shaped by host factors. And I
Right.
think that, it kind of undersells the strength and the robusticity of biofilms and
Yes.
how absolutely incredible those bacterial to bacterial bonds and associations are, but it absolutely is shaped by the host, right? Like we can't ignore that and we can't assume
Right.
that that's not going to play a role in this. And so our strategy has really been to make sure that we have a very diverse pool of donors, right? That we've got somebody well suited for that and that we continue to curate that, right? And we continue to build on that. So we have a paper coming out we call it our super donor assessment tool. We need a much better term for that.
Yeah, right.
Like SDAT or something. I don't know. We need a better term. But the whole point of developing this algorithm in that paper was to really figure out how do we identify the best donor given a particular set of circumstances. And so if we know somebody has a particular oral pH, or if we know somebody has less or more saliva production, or whatever that looks like, we could then effectively find a donor that might fit that person and, hopefully fit that. We still got a long way to go in sort of finding, I would say, a diverse enough pool of donors. But
Right.
that's one of the hopes that I would hope we can get to. You always hope that like, yeah, you're going to have one magical donor that's just going to work for everybody. And in the FMT field, they do have these people called super donors, right? These people that work really well, and they don't really understand right now why they work so well. And I would argue we don't understand those bacteria to bacterial relationships well enough to understand why they work well,
Agreed.
because they work in diverse sets of recipients.
Right.
And so there's something, quote unquote magical, right? Or super about, about that donor's microbes. And I think we have the same sort of effect in the mouth to some degree, but I think we can't be, ignorant to the fact that there will be probably some recipients that a super donor doesn't work for, right? Just as there are in the gut. And so how do we try to like future proof that? How do we plan for that? Yeah, because I would say like I would love to think that we could have a study with 50,000 Americans and we had all of their own genomics and all of their, physiological differences of their mouth and like all of that information that we can put into all one perfect model and be able to understand, okay, this microbe ties to that human variant or this whatever, and figure that all out. But without proper NIH funding to say the least, right? We're a long ways away from that. And so in the meantime, do we not try to push forward with transplants because we don't fully understand that if we know that the transplants still work? I think we still push forward and we expect some rate of failure and we try to do our best to match donors and recipients to try to avoid some of that failure if possible. Does that make sense?
Absolutely. Yes, absolutely. I mean, we've come a long way in vaccinology and that started hundreds of years ago, right?
Exactly. We've got to start somewhere and we've got to start collecting data too on what it means to put different donors in different recipients, right? And see what folks do well with which transplant and not, right? And feed that information back into the model and continue to iterate and develop better transplants.
So another factor that was brought to light when I was listening to some of your work previously, and that is that, we've all been using fluoride forever and ever and ever. And so there's evidence that possibly that integration process of utilization is now in our teeth. And I don't know what the half-life of that is. Like, does it ever go away? If we stop using fluoride, is it then gone after like a couple years? But the question is really like the durability of fluoride, if you have any thoughts on that. But more than that, when we're looking to utilize this type of therapy, but yet we've been kind of preemptively strengthening our teeth with this fluoride molecule. How does that impact this opportunity?
Yeah, that's a really great question. I love talking to you guys. This is fantastic. So from our perspective, we are also at a turning point in the United States where fluoride is being pulled out of water systems. I mean, even locally here where I'm at in State College, Pennsylvania, we're losing our fluoride.
Yep.
And so, we need to have better tools that can, again, try to future-proof this, try to think about how we get around it. I don't remember what the half-life of fluoride is. So that's a really interesting question. I was like, I could Google that quickly and figure that out. But
well, I think once it's been integrated, I don't know what the half-life is. I know what it is out like free, but
sorry. I mean, like
once it's integrated,
once it's there. And I think that that's a really positive thing, right? Like that's amazing. That's phenomenal, right? Like, let's not downplay the effects of that having fluoride integrated into our teeth and building stronger teeth, right? I think that's super important. But one of the other factors is that fluoride is also very antimicrobial, right? And one of the arguments for why we have this industrialized disease-associated microbiome today is because we have been hitting it with antimicrobials for decades and decades. We see a huge big shift after the Industrial Revolution. And we thought like, oh gosh, that's probably mostly just sugar, but it doesn't explain all the shifts from the Industrial Revolution till today. And so now we have, studies going clear up until the 1950s. And we think it's actually a lot of this antimicrobial rhetoric that comes along, the same time that everybody starts using soap and all these household cleaners. And it's really this like clean mentality post-World War II. The same thing happens in our mouth, right? We have the first sort of more commercialized toothbrush in 1938. We've got then these very antimicrobial toothpaste. They, eventually start adding fluoride and things to that with them, but they're still like very astringent, very, like kill the microbes, right? That's the messaging, like kill the bacteria in your mouth. But these microbes are so adaptive, right? They didn't die. I mean, some of them did, right? For sure, we've lost species in our mouths over this period, but at the same time, they just became more resistant to those things, right? And so now you have effectively bad bacteria. I don't like using that label, but you can think of it
different. They're different.
Yeah. That's great. So, you have these bacteria in your mouths, right, that now are capable of kind of evading and avoiding some of this stuff. This is why we have to brush our teeth all the time. This is why we have to keep continually using these products to see these effects and then still have them removed, right? And so, So I think from our perspective, it's like we've gotten ourselves into this situation where bacteria are very kind of adaptable to fluoride. And it's one of the things we're really interested in studying with indigenous communities who are, they're not absent, but are more devoid of sort of using industrialized toothpaste and things like that, is to understand some of that kind of fluoride effect on the microbes in the mouth.
Yeah.
But I think for most of the technology development for OMT, we're kind of making this assumption that everybody has some level of exposure to that. And, we need to have microbes that are, again, kind of born and bred in that environment, right? And can be robust
Right.
in that environment, like regardless of what's going to happen, if that makes sense.
Yeah.
And so yeah, all of our donors thus far are from industrialized areas. They may not have been born in industrialized areas, but they're living in those spaces currently, kind of with that antimicrobial assault on them.
Amazing. Again, I love that your research is so thoughtful and deep in that thoughtfulness. So with that in mind, when you're doing your model to kind of continue on and propagate these communities, these healthy communities from your donors, how many times can you go back to that sample and amplify? Does it persist? Like, does the same community persist over time, over multiple amplifications?
Yeah, so that's a great question. And that's something we're actually in the middle of testing. So
Okay.
so we know we can do at least sort of 3 or 4, and we still see largely the exact same microbial communities. When we take a fresh sample from someone, though, and we grow it in our flow cell, we do lose about 15% of the microbes.
Yeah.
And those are microbes that are just not well adapted to growing in this new artificial mouth, right? It's much more oxygenated, right? We don't have many more anaerobic pockets. So a lot of those anaerobes, those microbes that don't like oxygen, they keel over straight away and they don't come back, right? And so, it's an interesting kind of experiment to think about how that translates to the human mouth. But it also means that there's over 200 microbes for most donors that are very happy to live with each other in that environment. And they're very reproducibly grown. And they will grow very happily for at least 21 days in that environment together.
Wow.
So as long as there's fresh nutrients coming in, you can kind of think
Right.
about it just being the mouth, right? They're very happy to just keep living there and living in that biofilm environment. And, there's sloughing, there's some that die, but new ones grow and replace them. So I don't know. It is kind of this like bizarre thing to think about how we probably aren't recapitulating we're not recapitulating everything in someone's mouth. But it's enough of a stronger sort of signal, we think, to override that existing one, because it is still over 200. And in fact, some of our donors that we use for a lot of our testing, it's 230 different species that we're transplanting all the time. So, they're very happy living with one another. And it's not all of them, but it's a lot of them. So I don't know if that answers your question, though.
Yeah, definitely. Absolutely. It just makes me want to I'm sure you're always iterating. You already said you're on type 3 of your model. So I'm sure you're already kind of thinking of how you could create a crevice of low oxygenation. And that's a fun puzzle to be in.
It is.
Very cool. So you have already done animal models in cavities. So I love that you aren't relying on the gavage model. That just irritates me to no end. And we can get into why, but can you give us a take home of what you learned with your animal caries model?
Yeah. So the big take home is that in any of the animals that received an OMT from any donor, they never got severe decay, even under a model where we were doing this in rats. So we're feeding these rats sugar 5 times a day. They become highly addicted, right? They love sugar. Like me or anything else.
Not alone there.
Right? You feel for them. So in that model, all of our rats who received an OMT, no one got severe decay, whereas the rats who did not receive an OMT did get severe decay. So from that perspective, we think it's protective against severe disease. Now, even in what we would call moderate decay or moderate caries, and so this would be a score of 1, we see a 55% reduction in I always want to call them mice, but they're rats. They are rats.
Right.
Just rodents.
Right.
They're rodents. We see a 55% reduction in even sort of this mild to moderate initial phases of decay in the mouth. So, that for us is still really exciting. Does it completely block it from ever happening, never, ever, ever again? Absolutely not, right? These are still sort of living organisms that are going to do their own thing. And they're going to meld with the recipient's microbiome too. So that's one of the other things we've learned from this this rat model. I did it again, rat model. It's one of the things we've learned from that, though, is that, the end result really is kind of a unique microbiome of those donated microbes plus the host microbes at the same time. And so, we're getting protection against severe decay, which we're really, really excited about. It means this technology, could really be solutions for people that are doing absolutely everything they can and they still can't keep
Right.
cavities from coming, right? This could be a really great preventative solution for them.
Very cool. Okay, so I know you're going into the perio model, and then you're moving to mice, where it's more defined, I would say. I know.
Right.
So, what model of perio are you using? Don't say gavage.
I was going to say, let's have this conversation. Yeah, we are giving them P-Ginge to instigate that. We wanted it to be a more microbially focused version rather
than the ligature, which is just inflammation.
Yeah. Exactly. Yeah. I mean, this was something we discussed and we went backwards and forwards on it, and we had reviewers say, well, if you use the ligature model, you would at least have very reproducible sort of inflammation from this and you could test that out. And yeah, I don't know. I think we consider though, our technology is probably really fighting against the sort of overgrowth of P-Ginge, right? That if you have these host microbes, that they would be able to control P-Ginge growth in some way, right? Or dampen its effects. And so we really wanted P-Ginge in the mouth and we wanted it to be there in that mouse causing both local and systemic disease to see if we could recover any of those issues with an OMT. So yeah, I would love to hear all of your thoughts on gavage and all of that.
So for those of you outside of the field and just, listening in as a hygienist and dentist, one of the most commonly used models in perio research is the mouse and this gavage model, the Baker model, we call it. So you give the mice a large bolus of Porphyromonas gingivalis daily for several days, many days, and it actually takes 2 weeks for it to really start showing disease, which begs the question, is it really that initial issue in the mouth, or have you just disrupted the guts and now you have systemic inflammation and that's what's causing? So there's all sorts of postulation, and there is some great research that's been done to show that yes, there is disruption to the gut. Yes, it does go systemic. We have inflammation. So it becomes this very messy model, made worse by a high-fat diet, by the way, a Western diet, we call it. And it gets more and more complicated the more you talk about it. But I know in your research specifically, you are going into the guts and you are kind of doing this further inquiry. So I think kind of understanding where you're going with that, I love that you're going to be the one probably that marries the oral and gut for us in a really meaningful, applied way. So tell me all about that and your thought process.
Yeah, this is exactly it. And so we are not shying away from that gut effect. We actually want to understand that more.
Excellent.
And so we collect, and even in this previous model in rats for caries, we also collected gut samples.
Yes.
We were very curious to know how changing things in the mouth might influence the gut.
Do they?
Yeah. So we're not shying away from that. We lean into it. And, we are very much aware that this systemic inflammation could be one of the things that, allows gum disease to happen, for lack of a better description there.
Yes.
And so absolutely. So in this particular mouse model, we are taking fecal pellets, both, before and after transplant, but also on a weekly sort of basis after transplant. And then when we get to the end of the experiment, we sacrifice the mice. We also take histology samples from the gut all over. And we also take a bunch of oral samples, right? That makes sense. That's what we're trying to look at.
Of course, yes.
But we're trying to marry this with what's happening in the gut at the same time. And so that is really one of the focuses, right? If you can also take something orally that's very easy to get into your mouth and have positive benefits on your gut flora, your gut microbiota, we want to know about those, right? And that could be a really sort of awesome new thing. So for us, we kind of leaned into that model. But I agree very much in the literature, if you don't know what's happening systemically and you're just focused in on the mouth, you don't know what you've kind of altered or where you're fixing things or not fixing things, right? Feeding in together. We did a little bit of probiotic work together. That was actually how Peter Zellin and I got to know each other at the University of Adelaide. And it was working on periodontal disease and a probiotic. And we also did the gut and the mouth together at the same time. And so yeah, for us, it's more, there probably is this systemic mechanism that we need to also just be aware of, as well as the local mechanisms. And it's about bringing those two together and sort of tying them together that we're really after. For us, it also becomes
amazing
of safety too, right?
Yeah.
If you're going to take an oral microbiome transplant, you want to make sure it's not causing chronic inflammation and, systemic, like crazy inflammation in your body. Like you want to know that like systemically you'd be healthy as well as, your mouth or whatever. So for us,
Or conversely, if you do have, some IBS or inflammation in your guts, that you could potentially be improving by improving the oral health and like keeping an eye on that and making sure that we have data to support those future examinations as well. So I deeply appreciate this connected research.
Yeah.
Because it's just it's one donut hole.
I love that description.
Yeah.
We can't separate the two.
Bless you too.
So speaking of one donut hole, to rule them all. Yeah, I went there. We know that host factors and environment play a role, but we also have to consider that environment means diet and sustaining some of these long-term benefits. Do you think that altering healthy diets is, I think we all want a healthier diet, but for some of us, it's harder than others. So it becomes this kind of ethical discussion at the end of the day, like how much can we actually expect somebody to alter their behavior and diet when they may live in a food desert and be socioeconomically challenged and so on. But can you speak to some of the diet components of this research with those pieces in mind?
Yeah, that's such a great question. And I appreciate your approach to that and the way that you asked that question. So I'm also a professor of bioethics. And so we think a lot about the ethics behind what we're doing and also how we make this technology accessible to everyone. And that's all kind of the motivation behind all of this work. And we had an awesome PhD student, and now she's a postdoc working for Lucy. Her name is Sonia Nath. And she's done a phenomenal job at characterizing these healthy donors, but also in our work with indigenous communities too, also understanding how these socioeconomic factors, especially as they contribute to diet and access to healthcare, also shape these microbial communities in the mouth. So absolutely something we're aware of and something we're trying to think through and again, future-proof as we plan out this new technology. The short of it is, diet absolutely is influencing the microbes in your mouth. This is something I think that it's also very bizarre though, and how it got manifested in the dental field and then how we think about it from a microbiology perspective. The first paper we published on ancient human oral microbiomes came out in Nature Genetics in 2013. And we had a horrible time trying to get that paper finished and get it accepted for publication somewhere because we could show that there were dietary changes in the microbiome. And all the reviewers were people who worked on modern industrial oral microbiomes. And they just said flat out, the diet does not change the oral microbiome. It does not affect it. That was the dogma in the field in 2013. Diet doesn't influence the oral microbiome. And we're just sitting there like, oh my gosh, of course it does. It's different nutrients and different microfields. From the microbiology, it makes complete sense that would be the case. But from the dental field, it was not, right? And so we had to fight against that hill. And I remember presenting our early work too, just talking about how diet influences the microbiome at, the International Association for Dental Research conference in 2015 or something like that, and getting many questions about how, well, your research can't be true because the diet doesn't influence the oral microbiome.
Oh,
so
my Lord.
We've come a long way. We've come a long, long way. And now we have, numerous papers, and there's several more that'll come out here shortly that show that, effectively sugar is just this thing that absolutely completely transforms the oral microbiome. So if you have access to large amounts of processed sugar, your oral microbiome changes. And it changes because it's an amazing food source for many of those, microbes in your mouth, especially the ones like strep mutans and other streps that like to cause decay in the mouth, right? It absolutely does transform that. But if you're looking at people who don't eat large amounts of industrialized processed sugar, then it's other sugars that now start to transform the mouth. Right. Some of those fruit-based sugars, right, that can transform it. And it's not necessarily in a bad way. It just has an effect on the types of microbes that are in the mouth, right? It's still somebody who's orally healthy, but it's a different type of sugar that might actually play a bigger role in shaping those types of microbes. So it's a very complex process, I guess, is the short answer, right? And how diet sort of shapes these types of microbes and how we might be thinking about that in the context of transplantation. So one of the amazing things, though, that we did when we selected our donors for transplantation was that we didn't shy away from people who ate sugar. We said, you want to eat 300-400 grams of sugar a day and you still got great teeth.
Yes, I do.
I really want you. Yeah.
Yes.
You have great teeth and you eat that sugar? Like, yes, that's absolutely, you are amazing and we want you.
Jonathan, there's a donor for you.
Yes.
Let me know. Sign me up. Next
I'm
study.
just so you know, I have a problem as well. So
Oh my gosh, yay.
I'm holding up gummy lips that
Right.
are definitely sugar.
That's awesome. Yeah. So, I mean, some of our donors are people who still eat. We have whole dietary records of them, right? They still eat tremendous amounts of sugar, yet have perfect
Yes.
oral health. And so I think, it's not that's not all host factors, right? That shapes that. Some of that is what bacteria you have and how they metabolize that sugar. And when they metabolize that sugar, what are the byproducts of it? And can those byproducts be controlled? Right. So, for example, is acid production as a result of that sugar, can it be controlled by a different microbe? Can another microbe use that acid in some other way and neutralize it? Right. And so I think that's a lot of the premise of what we're playing with, is that we know where our technology is going to be put into people who are eating lots of sugar and not have access to, healthy, nutritious foods all the time, or people who flat out just don't want to eat healthy, nutritious foods for whatever reason.
Right.
We know that that's going to be the case. We expect that to be the case. And so, what can we do in donor selection? What can we do to help our in vitro model grow the right types of microbes that can adjust for that and can account for that, so that when we're putting them in folks who maybe don't have control over the diet for one reason or another, that it's still an effective technology. Does that make sense?
Yes, absolutely. Much like sugar, did you have healthy mouths for comorbidities?
Yeah, so we did not take, this was something we were really concerned about. So we didn't take donors from anybody who had any active pharmaceutical consumption for a
Oh, sure. Yes.
And
that limits the comorbidity issue. Sure.
Yeah, it does. But it doesn't get rid of it completely. And it's something I think about a lot. So in the FMT field, there were a couple very early transplants where people got comorbidities later because of the transplant. And so there's a very famous study between two sisters. And one sister was
Yes.
lean, one sister was obese, and the lean one got a transplant from the obese sister, and it cured the issues and the fecal transplant took, and that was really awesome. But then she later became obese. And there's a couple other examples of this where, there were lawsuits, and I mean, there's like, if it's a whole rabbit hole before bedtime when you want to go down, like there's all sorts of very entertaining stories to read in that space. But these not understanding maybe what these comorbidities could be, I think it's really critical for us to be thinking about that. So yes, we don't have donors who have those comorbidities, but our donors are also, in many cases between the ages of 20 and 30. We don't know if they're going to develop Alzheimer's when they're 65, right? Because of maybe something in their mouth. And so I guess from my perspective, it's all about folks who would want to use our technology, weighing the benefits and risks of, do I have, for example, severe enough decay that I absolutely need something to get rid of this because I want teeth in my mouth, right? Versus, I'm worried about getting Alzheimer's when I'm 65 or something along those lines, right? Understanding that there's a lot of environmental and social and genetic factors that also play into getting those diseases later in life that probably they're contributing probably a much stronger signal, I guess, for lack of a better term.
Right, exactly.
In that space. But yeah, so our donors, we absolutely did kind of flag for that. But it's something that keeps me up at night, just making sure we're doing everything absolutely to, the best of our ability to make sure that this is safe and healthy for people to use.
I guess I'm still wondering if it is possible, once you have systemic inflammation, to maintain a healthy mouth.
It's a good question. I know in many of the systemic diseases that we wrestle with, and many of the studies that we do with Indigenous communities, a lot of what we're looking for is how the oral microbiome plays roles in those systemic diseases.
Right.
And we see a lot of association. And so, that research is all still ongoing, but we know that there's connections there. And so you can't be oblivious to them, right? Like you've got to hit them head on, and we'll do our best. I always think about it, if I wasn't willing to put this technology in my own body, would I be willing to make sure it's available for somebody else? And so that's a lot of what our team thinks about, and a lot of how we approach it is making sure it's a technology that we would also be very happy to use and, know as many of the consequences as we can.
Right. I'm deeply fascinated by the role of our microbiomes in allergy and defense. So a lot of my work in my thesis lab was on the LPS structure and how it changes during pathology.
Awesome.
So the plaque samples will shift LPS phenotype, and those phenotypes are often exploited in vaccine technology, but also allergy as well. So there are all sorts of therapeutic interventions with these different types of phenotypes, but I think of using OMT approaches for that opportunity as well, minimizing allergy or amplifying defense.
Oh my gosh, that's a really fascinating idea. It's not something that we're currently exploring, but I will say, we've set out initially to do things like cavities and gum disease because I think those are kind of the obvious targets. It makes the most sense to, I think, explain to people about how you use this technology. But I will say we have a whole laundry list of other technologies coming, or other applications for this technology coming right behind it. And so exactly as you're mentioning there, right, like that could be a phenomenal sort of, additional application as we get further down the road.
Well, since nobody wants a vaccine, how do we bolster that defense mechanism, right?
Absolutely, right? Yeah. And this is a, fully non-toxic, all natural way to do that.
Right, exactly.
As weird as it is.
God, I love having you today. This is amazing. I could go all day and maybe take a pay cut and do a postdoc.
It would be great.
That'd be amazing. Jonathan, I defer to you. Do you have any follow-ups?
I'd love to hear, how has your research influenced your lifestyle choices?
Oh, nice.
Yeah, it's such a good question. And I think every time I give a public talk about my technology, and just the research in general, usually people come up to me and be like, so do you still brush your teeth? Like, or did you just stop doing that altogether? And, like I just tell people like, no, I have an industrial oral microbiome like the rest of us. We still have to brush our teeth because we're in a horrible state now, effectively. If I had a Neanderthal microbiome, I would be not brushing my teeth, but I don't have that. So, no, I think for me, a lot of the thinking has shaped kind of thinking about my kids and thinking about, I have two children, Leo and Bernie, who are 6 and 8. And, you're thinking about their future and kind of what you're doing now, how that might influence the types of microbes they have when they have their full adult dentition or when they have those things. Neither one of them have cavities yet, but, it's like we try to, lay off things like the sugary sodas and the, simple sugars because of a lot of my research, because I know that feeds into driving a lot of that. But at the same time, they're kids, right? They're going to love, they love candy. They love all those sort of things. But it is this kind of, being stuck and torn between the two. And same thing with myself. Like we, for the most part, really try to just avoid sugars because I've seen what that, kind of what that's done to our microbiomes. And I had, some industrialized grandparents. And so I know that I'm already screwed. I already have, this sort of associated microbe that the majority of people have today in the Western industrialized world. But at the same time, I also think a lot about populations where they don't have all of those microbes yet, and that we do have an opportunity here to either provide recommendations on how to avoid those sort of post-World War II, we call this the Great Acceleration, things that happened after the 1950s. How do they avoid getting those microbes? How do they avoid that sort of evolutionary process that happened to many people who lived in Europe? Like, how do we avoid that happening to them? Yeah. You don't want to be paternalistic or, anything along those lines, right? But if communities are up for that and they want those recommendations, like how do we get them that information and how do we collaborate with them to get them that sort of research? And so a lot of my thinking is thinking about these foods that are natural foods or foods that we wouldn't typically eat anymore in industrialized societies. And will that sort of bring some of these microbes back? One of the coolest stories that we've come across in some of my research was in Aboriginal Australians living in the Central Desert. They actually have a microbe called Microbiome. And the only other place that microbe is really found is in termite guts.
Ooh.
But it's in only Aboriginal Australians from the Central Desert. If you look at Aboriginal Australians and Torres Strait Islanders from up north, they don't have that microbe. And so it suggests that there's this connection to country, that there's this connection to diet that brings these microbes in. And, now with industrialized food sources, they're in a, they're not benefiting us, or maybe they are, we don't know. But I'm thinking a lot about those sort of natural food sources and the types of microbes they bring in, both to my oral microbiome, but also to my gut microbiome. And how do we bring some of those foods into our lives rather than shy away from them?
Right. Wow. Very cool. Nice.
It's a weird thing, I guess, to kind of end on is like, oh, you could eat termite and, improve your oral microbiome.
I mean, but if they're sitting in your walls, you might as well do something useful with it. God forbid.
Yeah. Yeah. Yeah,
Wow.
we still have so much to learn though about all of these microbes and what they do in our mouths. And we need to get out of
Truly.
working in European industrialized populations to understand that effect.
Yep.
Well, thank you so much for being here today. One of the things that we like to follow up with is there anything current day or literary that you're into in your, in all of your spare time?
Oh my goodness, in my spare time.
I know. What is that?
Yeah, I was actually thinking, gosh, this is super nerdy, but I've been going back through old school dental textbooks, like some of
Ooh, I love that.
the first ones that were made, and reading them just to understand how much our thinking has changed, like even in my lifetime, from like before I was born versus now, and our understanding. So that's kind of one of the nerdier things I've been doing, but
That's fun.
I'm getting ready to go on sabbatical, and we're going to start some new projects in Mongolia and Papua New Guinea. So I'm trying to learn a lot of that language and learn more before I get boots on the ground and start working with communities there. So I'm really excited about that too. That's kind of been taking up some of my personal time.
I bet. Wow. I'm incredibly envious right now. That sounds so great. Yeah. Okay. Well, it's also hard not to bring forward any current events because I don't want to talk about them.
Yeah, same. It's only Christmas currently.
Yeah. Let's stay in the mouth, I guess.
Yeah.
Anyway, thank you so much for being here. I will be following up with you for ever and ever, just because I want to see where your research goes in the continuum and see how much we can learn from you in your current studies, but then, where you're going is just really going to push a needle, and I'm excited for you. Thank you for stopping by today.
Well, thank you so much for having me, Camille and Jonathan. It's been really fun, and I loved all the questions you're asking. I'm going to be wrestling with them. Thanks so much for having me. I really appreciate it.
It was a pleasure.